Welcome to the RAD-BIO app. You can use the app within our website. There's a few options below, like the access to the glossary of technical terms. All these buttons take you directly to the data visualization dashboards. Let's launch here into a new tab for enhanced user experience. You can select from the different dashboards and provide more metadata by changing the way these options are listed. If you're new to this tool, let's have a look at the tutorial. It teaches you how to navigate a dashboard. There's two main sections. Along the top, you see there's a navigation section, so you can move around, and then there's this button that makes the different figures go large, and you can focus in on any one of the different types of data. And there's this green button in the bottom right-hand corner, and you can use that to download any of the data that you get interested in. So here's an example of how you can use the app to find some studies that have similar amounts of radiation dose as the journey to Mars. You can put filters on. So we're selecting filters here along the y-axis to narrow the range of radiation to be something comparable to the journey to Mars. And then we can use the app to look at the metadata related to that study and find the GLDS codes. These codes are the Gene Lab data study codes that allow you to access the primary data. And we have an app that allows you to visualize that on our website. So another way to navigate is this drop-down menu in the top right-hand corner. And so we're back to the beginning. So let's have a go and explore the app. This is the first dashboard. So this is the average total absorbed dose dashboard. There are these buttons, so again you can select a maximum amount of radiation because there's a lot of studies here that we're looking at. And you can use the pie chart to select the species of interest, so maybe you just want to filter down to look at the mouse studies. Let's go to the next dashboard using the navigation panel. So here there's many different types of radiation that we can look at, and many different radiation sources. Some from low Earth orbit in space, but also some terrestrial sources of radiation, such as ion particles being fired in a particle accelerator. So I'm mostly interested in the space-related radiation. So we can filter down through the data set and try and find some studies that provide information about radiation in low Earth orbit. Let's have a look at the next dashboard. We still have the filters switched on, so I'm going to switch those off. And now what we have here is a spaceflight related radiation visualizations. On the x-axis we have time, on the y-axis we have absorbed radiation dose, and so the longer you spend in space, the more radiation you get. There's correlation. And there's different vehicles that have taken different organisms up into orbit. You can kind of see what different types of methods, what sorts of transcriptional or proteomic types of profiling have been used to observe the organism's response to the spaceflight environment. You can zoom in. This study here has spent about 250 days in orbit, which, depending on when you go, is comparable to a Martian transit. But this Shenzhou's Arabidopsis study is a bit of an outlier. It was a relatively short period that it was up there, but during that time it received a relatively large amount of radiation. So finally, we were able to find another way to put this data in context, and that's by looking at the lunar orbiter, crater weather radiation monitoring system. So this is the lunar average, and what we're going to do, I'm going to have a look at the last 10 years of space radiation data as measured by the crater lunar orbiter. So here's 10 years, and every now and then there are some peaks, some moments when we have a large fluency of radiation coming through. So we can observe these radiation maxima, and we can also, for this 10-year period, calculate it the mean. And so we have integrated these data alongside the Gene Lab radiation measurements for the different studies which have been used to investigate life's response to radiation on Earth. And now we can essentially select the range of studies that have a comparable radiation load to either the radiation max or median en route to Mars. And then we can use the different species to filter down and look for, say, studies that used humans, or studies that use mice or Arabidopsis or other model organisms. There's many to choose from, and lots of people have their own preference. Once you've selected some studies of interest, it's good to get an overview of the metadata, the different factors that were being studied. And again, once more, you can filter through based on organism and search for your favorite model organism and the studies that were done on them. This allows you to focus on smaller subsets of data that have conserved metadata features in the original experiments. I'm a huge fan of the research that has been done on the International Space Station. So essentially we can filter down and have a look at the different model organisms that have been investigated on the International Space Station. If you find some studies of interest, again, you can always download these tables by clicking on this green download button, and then finally downloading here. You can save it as a spreadsheet or as a Word document. These data can be saved and investigated at a later time. I often like to take the GLDS accession numbers for NASA GeneLab and then access the primary genetic data or proteomic data associated with it. If you get confused at any point while using this app, we have a glossary of terms and have linked to some tools for converting different types of radiation dosimetry. There's also, in the glossary we also list the different types of abbreviations that are used by NASA during their astrobiology research. This work was performed in the Gilroy Lab as part of their NASA TOAST project. This was done in collaboration with the NASA Ames GeneLab project and the Jet Propulsion Laboratory MANGO project, which is part of the Interplanetary Protection Team's effort to help citizen scientists understand more about space biology.